Good morning.
Good morning.
Good morning.
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 Morning. Oh, you can't see me. Let's get started. Okay. Yeah, you look as awake as I feel. Don't look very awake. Okay, let's see. So you guys, I do need to let you know that the meeting is being recorded. How many of you have gotten on in the last couple days and tried to play the videos? Has anybody? Which is okay too.
I tried, but it didn't work.
Say it again.
I tried, but it didn't work. But it was a couple of days ago.
Have you tried within the last day or two?
I tried to, but it didn't work.
When did you try? When did you try?
Yesterday night.
Do you have a pop-up blocker on your phone, on your computer?
I think so.
Okay, if you have a pop-up blocker, you need to disable it.
Oh, okay.
Are you talking, which videos are you referring to again?
The lab videos.
The ones that are under the lecture materials?
No, the ones that are under the labs that we weren't being able to get to play on anybody's computer last week except for my own.
I can play them right now.
Okay, because I tried to copy them in the way another professor had them copied into her Blackboard site. It didn't work. That's the reason they wouldn't play last week. However, they should all play now. And if they don't, first, number one, try to disable any pop-up blocker you have on your phone, on your computer or on your phone. And then if that doesn't work, try calling IT technical support. Because if you haven't figured out I'm not a computer genius, I usually call somebody else when I need help with that kind of stuff. I'm slowly learning more and more. But if after you've called them and they can't figure out how to help you, then email or text me and tell me.
Anatomical directional terms.
Yep, that's the right video.
Sorry.
I just tried it and it worked.
Well, good. Okay, I'm glad. Yeah. So yeah, if there's a problem with that, try calling the IT technical support first and then give me a call.
Okay.
Because what I do is I try to figure out if there's a problem on my side because I can't fix your computer, I can only fix mine. And so all I can do is try to figure out if the problem's coming from my end. If you look over here, I wanted to show you something because I've gotten some questions about deadlines. And I am going to try to get them more and more uniform. Bear with me as I do that. But if you look right here on D2L, and I'm going to switch really quickly here. I think I actually am viewing this as a student. Yeah, I think it has me viewing it as a student. Do you see what I see when you go into Hack? When I go into 304, do you see the due dates on the right side of the screen for our class?
Yeah.
Yeah.
Okay. So when you have questions about when things are due, this is a helpful guide. It will tell you when things are due. When you do that virtual lab simulation, if you miss a step, it will not let you go forward. It will wait for you to figure out what step you've missed. So when it doesn't let you go forward with the experiment, the first thing you should check is whether or not you missed a step.
I tried to do the prep anatomy and physiology, and it was asking questions that weren't related to like the chapter we were reading. Is that supposed to be that way?
What was it asking related to? Do you remember?
I think it asked a question about sodium chloride or something like that.
That is going to be related in the long run.
Okay.
It isn't related right now.
Okay.
But when we start talking about nerves, it will be there.
Is that something that's graded? Because it's been like 6 years since I took any science classes.
Well, you're gonna love the next chapter. Yeah, that was a little sarcastic, but it wasn't sarcastic.
Right. Okay.
I
I
would
have
start
a question.
reviewing that material. Do you have like a basic chemistry book or even I would read your chapter really well.
Okay.
Because all physiology is biochemistry. We do cover some biochemistry in this class.
Okay.
I have a question.
Go ahead, Esther.
If you go under the quizzes, there is one that says chapter 1, reading. So I don't understand why that is coming.
This one right here.
Is it? No, I'm sorry. Go to grades.
I can't. You know what? I can try to go to grades here. I can't do that with you on here because I will show everyone's grades, which is a FERPA violation.
You see that chapter 1, reading?
Yes.
So, what is that?
That is the Connect reading. That's the Smart Book reading.
Okay. Because when I clicked on it, nothing came up.
Let me see. Let's see what happens here for a second.
But on my hand, nothing pop up.
It's a Connect. It's a McGraw-Hill Connect.
Is that the one if you go to, where is it at here? where it takes you to McGraw-Hill and it says 1 through 10. It's like a quiz.
Yes, probably.
So if you go, to your course home and then go down to chapter 1.
Just a sec. I'm gonna have you share your screen because I don't see what you see on Connect because I'm not technically a student. Bear
See,
with
I just
No.
tried to do that, but it's not highlighted like yours.
give me one moment. One moment, Esther. One moment. Let me get so I can actually allow sharing of screens. That wasn't it. Oh, I let it share screens. I know there's a way.
I have to hit it, I think.
You have to what?
Oh, you disabled it, yeah.
Yeah, so let me figure out how to not disable it. It automatically disables when I get on here.
No worries.
How did I do this before? Bear with me, I'll figure this out. Oh, all right, let me stop sharing. More. Stop. No, there was a way to do this because I've done it more than a few times in the last couple of days. Let's see here. Here. Okay, try to share your screen now.
Should I share it?
Yep. See if it works now.
All right. You guys see it?
Yeah.
All right. So when you go to course home and it brings you to this section here, and then it's this one right here. And it says launch. You launch it. Is this the reading quiz right here?
I'm 99%. Yeah, smart book on top. It is. I think it is.
So if we open this, are we reading the material or are we?
And then it's going to ask you some questions. It's going to click on it. Click on it. Because I'm going to be curious too.
Well, that's one of the things I wanted to ask you today. I didn't want to click on it because I didn't know what I was.
So you have a choice. If you look down here on the bottom, you have a choice where you can either learn more before you answer the questions or just start answering questions. If you have questions wrong, it's going to ask you to read something, I think.
Is this graded like on like how many we get right, or is like
It's graded on did you complete it or did
Completion? Okay. That's what,
I chose to read first. Is that like the chapter 1 from the book that we're supposed to read, or?
Yes.
Okay.
Pieces of chapter 1. Like it actually, if you think this is a lot of material, this is about a third of what is actually there and available that I've actually scaled back.
are you talking about your PowerPoint thing?
No, with the smart book assignments, when I go in to assign them, the 40 concepts right there on the right hand side, that circle is like 150 concepts. And because I don't want you to spend 3, 4, 5 hours on this, I scale it back. And so I tell it that I don't want you to have that many things. I want you to have less than that because I don't want you to put 5, 6, 7 hours into this. Does that make sense? Is that understood?
Yeah. So if we can go over here and do the start with the reading, and we can go through and read it on our own time, and then we can go, and it's just completion. We don't have to worry about.
You do me a favor, Josh. Click on that start with questions really quick. Just
sorry.
the other side. No, you're okay. Welcome to my world. You just do the other side.
When I did it, you could switch back and forth. It was just like trying to teach you the principles, and if you already read it, you could just like go through and answer the questions, but then if you like forget something, you can go and switch back and forth so you can read over the material to answer the questions again. It's really easy if you do it on your phone.
You know what, who's the one saying this?
Brooke?
For
Brooke,
this assignment,
will you make sure that it actually gave you a grade for it?
Yeah, I can look and see.
Yeah,
It's
just to
okay.
verify, because
I think you did, because I think I got an email, but I'll go look.
well, I know sometimes for some reason, it gets strange when you do things on your phone. And so I just want you to verify that it actually went through. And if it doesn't, screenshot, you guys, I don't care if you do it on your phone, in all honesty, but I need you to send me a screenshot if you go in and you find out that it does not work, that you completed it, but it did not give you a grade.
I
Because
did mine yesterday. It does grade you.
I did mine too, and I had a grade for this.
Yeah.
okay, so it gave you a grade. Did you do it on your phone?
I
No,
did
I
on
did
my
on the computer.
Okay, if you do it, if some of you decide that you want to do it on your phone, which is fine, make sure that you get a grade on D2L for it. Because if you don't, what I want you to do is send me a screenshot, either email it to me or text it to me. If you email it to me, I can't open certain photo documents on my computer. But if you text it to me and all I need to do is see that it says that you completed it, and I will give you the credit for it. Okay? Okay, so Josh, did that work?
What, to start with questions?
Yeah, well, let's just see what it does. Click
Okay.
on it.
And this is what it says.
It's okay to get questions wrong and you still earn 100% if you complete all the concepts by your due date. Submit your answers by selecting your confidence level. This will not affect your grade. So click got it there. Let's see here.
That's anatomy.
Click.
Physiology? Or wait, no.
Anatomy.
Anatomy.
Yep.
Anatomy.
Yep. So put in anatomy there. You guys are smart. You're figuring this out. What's your confidence level? Is it high, medium, or low at the very bottom there, Josh?
Whatever.
Okay. So next question. And it obviously is asking you about whether it's uppercase or lowercase. Don't worry about it. Taking a pulse by feeling the radial artery runs down the radius bone, is an example of what of the following?
Palpation.
Palpation.
So click palpation there, Josh. What's your confidence level? Okay. Next question, that's correct. It tells you at the bottom. The term for the process of carefully separating the tissues to reveal anatomical relationships. Josh, how about I tell you the answer? It's dissection.
Yeah, mind my spelling. It's probably terrible.
Well, you know what? I think it will still, if it's wrong, we'll figure it out. Put one more S in there. One more S in there. Okay. Correct answer. Okay. So that's how you do it. Does that make sense? And if you want to do some of the reading, you can. I don't know what it does. Do the next one. Go next question really quickly here. Most exploratory surgery has been replaced with, do me a favor, let's intentionally get one wrong here. Click dissection here. Probably a good answer to say low. So do you need help, or do you want to just go forward? Next question. It's medical imaging because no, we don't cut into people if we don't have to. You want to go forward to the next question? So it tells you whether or not it's wrong, right or wrong. Does that work? Does that make sense, what you're going to do?
Yeah, is there a way to go back to the last question before you?
I don't know.
And is there,
It's not going to,
can we save it and exit? Is,
exit assignment right there.
So if you exit it, you can get back on where you started. If you miss a question, you cannot go back to it, but you will get that question again.
oh, you'll eventually get back up to it? Will
Yeah.
it make you re-hit it? Okay.
Okay. You guys, for everyone who, that was a good demo, Josh. I appreciate you doing that because I don't see what it does either. I've just heard a little bit. Does that work for everyone? Do you understand how that works?
Yeah, I did mine already, but like I said, if you go over to the content where you have the grades, the chapter 1 reading, I was unable to highlight that to open it for.
It didn't allow you to open it?
No, just like you did, I was unable to do mine.
I wonder if you have you disabled the pop-up blockers on your phone?
Yeah, for this one, yes.
I would call the IT technical support there at the school.
Yeah, I completed this chapter too, but chapter reading, like chapter 1 reading, is still blank in the grade book for mine.
Exactly, yeah, that's the same thing I'm saying.
Yeah, I have the same thing. In the grades, it has chapter 1 reading, and it also has chapter 1 major themes of anatomy and
Yeah.
physiology.
Yeah,
Right.
Which the major themes of anatomy and physiology is complete, and
yeah,
the reading is not.
exactly. So
Yeah,
it's
same to me too.
yeah.
Give me until the end of so you're seeing it twice essentially.
The reading is blank, yeah. The chapter 1 reading.
Chapter 1 reading's about 3 down, and then a little further down is this one.
Right here. No, not this one.
Yeah, mine for that one, it says 10 out of 10 and I did it on my phone. So I don't know if that's the grade for the reading assignment.
I'm pretty sure that is it.
Okay.
You guys give me until the end of the week to figure out how to fix that.
Okay.
And I will I'll figure it out.
All right.
It's kind of like the video links. I end up calling someone else and they tell me what to do. So bear with me as I figure it out. Okay, I'm gonna stop. Josh, stop sharing your.
Oh.
No, you're okay.
Yeah, sorry.
Well, welcome to my world. Don't worry about it. Let's see here. I'm sharing now. Phone calls. Into go. What was I? I can't remember where we left off. I'm going to go to a certain place and you tell me if we're past that point. Did we get to this place?
No.
I think we stopped right before this.
Yes.
So we were about right here?
Yeah.
Yes.
Okay. So how does one assess certain aspects of the body structure and function? First one is inspection. Looking at body surfaces. Is there swelling there? You can also palpitate for swelling, but you can see it most often if you look for a goose egg on someone's head after they've went back and smacked their head. A rash, chicken pox, palpitation, for example, feeling for the radial artery and feeling for a pulse. Auscultation, listening to body sounds to evaluate the functioning of certain organs, listening to the lungs or heart using a stethoscope. Can also listen to the abdomen. Percussion, tapping on the body surface with fingertips and listening for a resulting echo. Cadaver dissections, cutting and separating human body tissues to reveal tissue relationships. Comparative anatomy, studying multiple species to learn about form, function, and evolution. I picked this example specifically because here's the human heart. Here's what the heart looks like in an earthworm. And if you look here, and I don't know how small this is for you on your screen, bear with me, I'm gonna make this a little bigger. If you look here, there are 5 little rings that make up that compose the heart of the earthworm. And having not taken a comparative anatomy course, but looking at just how that works, because I know there's a vein that runs through here and there's a vein that runs up here. And that these rings kind of encircle that vessel. I'm guessing what they do is it constricts and relaxes and pumps the blood throughout the worm. So a little different in comparison to us. Another one, if you can't observe what you're looking for with medical imaging, exploratory surgery, opening up the body and taking a look inside to see what's wrong and what can be done about it. Much more invasive. If your alternative is if you've tried medical imaging and you still can't figure out what's wrong, that's when you're going to be doing exploratory surgery. Medical imaging, viewing the inside of the body without surgery. Radiology, the branch of medicine concerning with imaging. Example, what we've talked about before now, the X-ray sonography, CT scans, MRI scans, PET scans, and DSA scans. Histopathology, the micro examination of tissue for signs of disease. I use this as an example because this is a pretty obvious histopathological samples here. This is a blood smear. This is a normal blood smear where you have the red blood cells And then you have a handful or a couple or a few white blood cells in there. This is leukemia. So cancer of the bone marrow or cancer of the production of white blood cells, where we have a lot more white blood cells in there and not as many red blood cells. Cytology, the study of structure and function of cells. Ultrastructure, the view of detail under the electron microscope. This would be the same as possibly taking this, a much smaller square of this up here. Here's your red blood cells. Here's your white blood cells. So this little divot right here, that's a platelet. Gross anatomy, the study of structures that can be seen with the naked eye. Looking at the abdominal viscera. Do you remember from lab what viscera means?
Against organ?
It means against the organ. That visceral layer, meaning it's the layer up against the organ. Viscera by itself means organ. So this is, abdominal viscera means looking at the organs within the abdominal area. This right here being the small intestines. I think this right here, just looking at this picture, that's your large intestines. So histology, taking a very thin section of this same tissue and putting on a slide with a slide cover and looking at it underneath the microscope. So name the method that would be used for each of the following. Listening to a patient for heart murmur.
Auscultation.
Auscultation. Studying the microscopic structure of the liver.
Is it the anatomy of the liver
Look with the microscope.
Histology.
Histology? Yes, that would work. What about microscopically examining liver tissue for the signs of hepatitis?
Histopathology?
Yep. What about learning the blood vessels of a cadaver?
Comparative anatomy.
I would say gross anatomy because a cadaver is a human, so I would say gross anatomy. Comparative anatomy is looking at other species. Performing breast self-examination.
palpation?
Correct. Okay. So the human body is a complexity of emergent properties where organisms, meaning us, are composed of organ systems, and organ systems are composed of organs. And multiple tissues come together to form one organ. Hundreds and thousands of cells come together to form a tissue. Organelles come together to form cells. Macromolecules such as DNA come together to form organelles within the cells. and molecules and atoms come together to form macromolecules. With each of these, there is an organization and an interaction that results in higher properties and properties that weren't present before. An interrelationship among organ systems allows for human life. For example, here we have the digestive system that takes in nutrients. Nutrients get broken down and they're absorbed into the cardiovascular system. The cardiovascular system pumps that material to about where the liver is, about right here. Then it pumps it up to the heart, and the heart pumps it out to the respiratory system, which allows for the interchange of gases. Then the blood gets filtered through the kidneys, and some of the waste products that are, the liver would be about right here. The liver, some of the waste products also get sent down to the kidneys, and whatever is foreign or inappropriate in there, or nutrients that can't be absorbed at that time, get passed out through the urinary system and the fecal tract. So this interplay of the organ systems allows for life. Which of these is correct for which of these is a correct representation of the hierarchy of biological organization from least to most complex?
C.
How many of you agree with her? Sound good? That it's not C? That's right. Atoms and molecules are part of which level of organization in the human body?
Chemical.
So you said the last one was C or B?
Yes, it's C.
Okay.
What about here? Atoms and molecules are part of which level of the human body? And do you guys agree with Esther again?
What did she say?
She said A, chemical.
Maybe cellular?
I think she was right for chemical.
Chemical?
Cellular would be atoms and molecules that are now being made into organelles. And then those organelles are put into the cells. A structure made up of two or more types of tissues that performs a specific set of functions is at what level of organization?
An organ.
Yeah.
It's the organ. This right here is a concept map, and we are going to see probably more than a few of these this semester. It is a good way of networking and showing the relationships between the different organ systems. There's a word bank down here too. So for example, the muscular system functions in posture and And this,
Movement.
say it one more time.
Movement.
Movement. Yep, or locomotion or something. I'm not looking at the word bank down there. What is it? What does it tell us? I think it says movement. You're right. I'm sorry, Devin. You're right. So I think we're going to see more than a few of these. No two anatomical variation. No two humans are exactly alike.
I'm sorry,
Some
Bertha. Is there a way to double check all the answers for that? Is
if
there gonna be?
if you want to fill it out and send me an email of it, I'll look at it and see if it's right.
Okay.
Do you want us to do that?
I don't. It doesn't. It doesn't matter to me. I'm not going to quiz you on that. It's one of those things where I don't know. You're going to have similar things on exams, but not to that complexity, because that's all the organ systems right there. It's not going to be that many fill in the blanks. It may be more difficult than that, but it's not going to. I'm not going to give you a complex map that has 20 or more fill in the blanks. So some individuals lack certain muscles. Some people have an atypical number of vertebrae. You have, I looked this up the other day, you have 34 vertebrae. It's either 33 or 34. Not on average, not everybody does. You can have an atypical number of certain organs. And some people also can exhibit what's called situus inversus, meaning that what was typically on the left side of their body is now on their right. And vice versa. When I was a student at Idaho State University, one of my friends there that worked for the school had a daughter who was born without any asymmetry of her organs. And when they saw it in the ultrasound, so they knew that it was odd, and they knew that they were kind of expecting that she was going to come out, and planned on having major surgery on her when she was born. And knowing, the fact that her heart was in the middle of her chest and her liver was in the middle of her body, and her stomach was somewhere around the middle. And she was not born with a spleen. And she was born at Primary Children's there in Salt Lake. They planned it knowing that they didn't know what they were getting into when she was born. And she came out and was fine. All physical signs and symptoms showed that she was fine. But when they go in and they do ultrasounds on her now, and she's probably about 10 or 11 at this point, her plumbing and her vessel structure is very different. Because with us, the fact that our aortic arch goes off from our heart on the left side, and it goes down on the left side of our body, and her heart is in the middle of her chest. And so she doesn't have that same arch. She has two, can't remember if they call it two lefts of the heart. When you look at the heart, and I don't think we do it in this class, but next semester when you look at the heart, the left and the right side of the heart are very different in their anatomy. She has two halves of the same. I think what she has is two lefts of the heart. And it works for her. Her older sister was in the 90 percentile for birth and weight, and she is too. And for some reason, it's just, regardless of how weird her anatomy is, it's worked for her so far. And I honestly, in a lot of ways, I think that the human body is absolutely amazing in its development, because it will, if it is at all possible, it will figure out a way to make something work. And it will figure out a way to survive and make the plumbing work so that body slash baby slash fetus is functional. So back off my tangent there. Anatomical variation. Some people are born with horseshoe kidneys, where the two kidneys are connected. Some people, rather than having one on the right side and one on the left side, have one down in the pelvic area. So right here, here's the normal branching off the aorta. Here's variations that you can see that have been observed. There are basic functions that distinguish living from the non-living. One of them is a sense of organization. I put this cat here and I put the rock here because the rock is non-living. And the cat is living, and there's an organization to the tissue and the organ systems that allows for life. All living things are made from cells, and the cells can divide and replace one another. All living things perform metabolic activities to maintain life. They also respond to stimuli. We talked about the pain response last week. All living things are able to respond to stimuli. The fight or flight response. Movement. Homeostasis, keeping your internal conditions within a narrow range. Development, cell differentiation and growth. Reproduction, and being able to evolve. My one main argument for evolution is, I'm trying to think, I know we have some people here who have kids. I know that Jessica has a kid, and I know that Josh has a kid. I think Esther has some kids too.
Yeah.
And I only see certain people's pictures here, so be patient with me because I can't see you if you're not here on my screen. If you look at, Jessica, you have a 2-year-old son. You look at your son and compare him with your husband or his dad or something to that effect. What Is he exactly like one or both of you? Is he what? Does he look exactly? Is he exactly like you, or is he exactly like his dad?
No, I actually have 4 kids. My youngest is 6. But no, none of my kids are exactly like me or their father. They're kind of a mix.
They're a mix, and on top of that, there's genetic exchanges that occur before they even start developing. And so what you're essentially going to get is someone who's different from anyone else in the world, based on the genetic exchanges that occur even before the egg and the sperm fertilize. And that's the best argument I can give you for evolution, is that everything and everyone evolves. We evolve as a population, but individuals, everyone that is born is uniquely different from anyone else, just based on the genetics and the DNA itself. So cells are able to transport materials from one region of the cell to another, just as the body is able to transport substances to different locations, or skeletal muscles working with your skeleton can transport substances to different locations, or skeletal muscles working with your skeleton can transport you from the sofa to the refrigerator. What basic life process is illustrated by this description?
Movement.
Movement. Homeostasis is defined as the ability to detect change, activate mechanisms that oppose it, and thereby maintain relatively stable internal conditions. So the example we have here is we have a thermostat bear with me. We have a thermometer that measures the temperature in a room. When the temperature falls below 19 degrees Celsius or 87 or 67 degrees Fahrenheit, the thermostat is activated that generates some heat. The heat is put out, the room temperature rises to 20 degrees Celsius. At that point, when with that 1 degree change, the thermostat is shut off, and the room cools down again to 19 degrees Celsius. So this is a feedback mechanism to alter that drop, bring that drop back to 19 degrees Celsius. What is the definition of homeostasis?
D.
We'll say it one more time. D is in Delta or C is in Charlie?
D as in
Delta.
dog.
How many of you agree with her? Emma, you're right.
I agree.
Okay. So there are certain components involved in a feedback loop that maintains homeostasis. The 3 main components, which I'll explain to you afterwards, but let me tell you the individual parts of this, explain the individual parts of this feedback loop. So initially we have a variable, and the change in this variable is the one that's actually being monitored. We have a stimulus that produces a change in the variable to knock it slightly out of the normal range where it should be. So we have an imbalance in that variable. The change is detected by a receptor. So there's a receptor the receptor's job is to evaluate where this variable is. And when it changes, the receptor senses that change. The receptor then sends a signal to the control center saying there's been a change. The control center, this is also called the integration center, which is spelled integration is spelled I as in India, N as in November, T as in Tango, E as in Echo, G as in Golf, R as in Romeo, A as in Alpha, T as in Tango, I as in India, O as in Oscar, N as in November. So the controller, the integration center, gets a signal from the receptor saying that there's been a change in the variable. The control center sends out a signal to an effector output, sends out a signal to an effector that makes an action to bring that variable back to where the change back to where it should be. An example of this is when we overheat. When this last summer, today's probably the coolest day that it's been in a while, get really hot. Our vessels, to keep us from burning and causing permanent damage to our organs, they vasodilate at the surface. And this vasodilation, vasal meaning veins, dilation meaning an enlargement of the veins. So the vessels they go from being reasonably constricted to dilating out. And the reason they do that is to allow the heat that's in the blood to be released from the blood and be dissipated into the environment. So we get this vasodilation to drop the heat level in the body. And then when it gets below a certain point, like 98.6 degrees Fahrenheit, the vessels constrict again to start reserving heat and maintaining heat and not letting it go so much, not losing so much heat until we get above that point again, and then it happens again and the vessels dilate. Another example of this is someone rises from a bed. Gravity pulls on the blood, and the blood drains from the upper body, creating a drop in blood pressure in the upper body. This is an imbalance in homeostasis. If we drop blood pressure, we drop blood flow. And so it gets dropped a little bit. There are receptors in the right atrium of the heart and in the aortic arch. And I think they may also be in the carotid artery. I have to think about that one. Their job is to look at pressure. You guys, have you ever heard of a barometer? Jessica's heard of a barometer. I think Emma was nodding too. What's a barometer?
I think it's used to measure. I'm not sure.
What does it measure?
Pressure?
It measures pressure.
Yeah.
It measures atmospheric pressure. A barometer and a baroreceptor, this is a receptor that measures pressure. So it's going to measure blood pressure in this case. Those receptors, when they sense that drop in blood pressure, send a signal to the integrating center or the control center. So those two terms are the same. And the brain, the nervous system sends out a signal saying that the heartbeat needs to go up and the vessels need to constrict to bring the blood pressure back so that we have regular flow to the upper body again. And the blood pressure rises to normal levels. There are two different control or integrating centers involved in maintaining homeostasis. One of them is the nervous system. It sends nerve impulses via action potentials that can cause rapid changes. The endocrine system, which secretes hormones from glands into the blood, and it acts a little more slowly. These systems can act together or independently of one another. Nerve impulses tend to be very quick. Hormones have to be secreted from glands, then they have to travel in the bloodstream to wherever their effector organ is, wherever whatever they're supposed to affect. So it acts more slowly, but it is a more prolonged response. So nerve impulses, it can be very fast, and it can and then you come down really quickly. The response occurs and then it stops. Endocrine system, you secrete the hormones, it's a little slower, but then we have to, with the response, we have a breakdown. You have a breakdown of hormones that has to occur before that response actually goes away. And that can take time. So it's a delayed response, but it occurs for a longer period. Once the response is there, it's going to be there for a while. So up to this point, we've talked about negative feedback loops where a change happens and it's sensed and the signal is sent out from the control center, and the control center sends a signal to an effector that says, put that response back to where it's supposed to be. That needs to be within this range. It can't be out here, it can't be out here. It's got to be within this range. Positive feedback loop is different. One example of a positive feedback loop is the head of the fetus during childbirth. The head of the fetus pushes against the cervix, the neck of the uterus. And that pressure, all you get is that I've never had a child, but you get that pressure, that mild contraction where the baby's head gets pushed against that cervix. Receptors down in the cervix send a nerve impulse to the brain, to the pituitary gland in the brain, and the pituitary gland secretes a hormone called oxytocin. Oxytocin stimulates the uterine to contract even more, pushing the baby's head harder on the cervix. So rather than something that immediately brings the change back brings the variable back into the range it's supposed to be, in positive feedback loops, we keep pushing it forward towards the change that's already occurred. So this is a self-amplifying cycle where a change results in a greater change in the same direction. In the same deviated change, it keeps becoming more and more deviated until the child is out and that pressure is removed. My question for you here is, what are the components of the feedback loop in a positive feedback loop? What are the components of that the effector and the integration center and the receptor, the integration center, and the effector? The receptors right here, where the head of the fetus is, it's going to be similar to, I don't want to say they're baroreceptors, but a stretch receptor that's measuring that stretch of the cervix. So that's the receptor. Integration center is the pituitary gland in the brain that secretes out the hormone oxytocin. And the oxytocin stimulates the uterine walls, which are the effector, to contract more.
Can you repeat that one more time?
Of course. So the receptors are stretch receptors within the cervix. The stretch receptors send nerve impulses to the pituitary gland in the brain, which is the integration or the control center. And the effector, excuse me, let me back up. Just the pituitary gland secretes oxytocin, which signals the effector organ, which is the uterine muscles, to contract. More than anything, what I want you to understand here is, can you look at a positive feedback loop and determine what are the components of the feedback loop? Can you look at a pathway like this and determine what are the components of the feedback loop? What senses the change? Which in this case is the stretch receptors.
So when you ask us like a question similar to this or something, and you want us to identify this, when we are identifying it, as long as we do the receptor, the integrator or whatever, and the effector, those are the three that you're looking for?
Yes, I would probably ask you, if I was going to make this a question on an exam, I would ask you, I might make it a matching because we're doing these on using Connect. I would ask you to identify the components of the feedback loop here. And it would be a matching, so there's certain. Give me 1 second, I'm just thinking how I could ask this. Bear with me just for 1 moment here. I'm gonna go into something. On time. Computer's just being slow. Give me a minute. One. Obviously that didn't work. Let me try this again. Maybe.
I guess kind of my question is too, like, so a few pages earlier, when you're looking at the physiological example of maintaining homeostasis, when you have these loops like this, you're looking for like the four or the fives. Are you looking for all five? The stimulus,
I would
receptor, the
specifically
integration.
it looks at 3 components. It looks for the receptor, it looks for the integration center or the control center, and it looks for the effector.
Excellent. That's it. Thank you.
If I ask for something else, I'm gonna have to be really specific about it. I would have to give you a very specific.
You have a 6.
Say it again. I would have to give you, if I asked you, bear with me, if I asked you, I wish I could pull this up. I don't know why I can't.
I think you have a 6 after the.
Don't know why I'm getting blocked here. Give me a day and I'll figure out why I'm getting blocked here. Give me until Thursday, and I will show you an example of a question, okay? For right now, I want you to understand more than anything from this, I want you to understand that the same components that are in a negative feedback loop are also those same, the receptor, the effector, the control center. You can find those in a positive feedback loop. You can identify them in a negative feedback loop. For example, here's another one, and I'm gonna, if you look at this, A positive feedback loop. There's a tear in the blood vessel. Platelets that are floating along in the blood sense that tear in the blood vessel. They immediately start adhering to that break and start releasing chemicals. The chemicals attract more platelets to bind to that area. And platelets, and those platelets that bind to that area release more chemicals and attract more platelets until there's a platelet plug formed. When the tear is when there's no longer blood leaking from that tear, the platelets are no longer activated, and we stop getting an activation of the clotting response.
Excuse me, Prof.
Go ahead.
Yeah, I think the reason the address is not going through is you have a 6 after the www.
I'm not putting that in there, and that's the weird thing, is I'm not putting that in there. If you
No, you have a 6. You're not supposed to have a 6.
I know it's right there, but when I type it in there, I'm not putting it in there. It's automatically going in there. If you watch what I'm typing here, do you see it?
Yes.
So I put that in, I click it. You still didn't get that 6 up there.
Okay.
Yeah. Give me until Thursday. I'll actually, what I'm going to do with the chapter 2 quiz is I'm going to have some of those matching things. I'm going to have some similar things to what you will see on the exam.
Okay.
Which of the following is an example of a negative feedback system? I'm gonna zoom out just a little bit here. During labor, uterine contractions stimulate the release of oxytocin, which strengthens the contractions of the smooth muscle in the uterine wall. A decrease in blood pressure and heart function during severe blood loss. Formation of a blood clot or a scab after someone has received a cut to the arm. Scratching an itchy skin rash, which makes the rash spread and itch even more. Or none of these examples are those of negative feedback.
See?
Okay.
I think it is E. And the reason is because the formation of that blood clot is the same as this right here. Once you quit having blood loss, the signal goes away and it quits amplifying. But until you stop the blood loss and you stop the tearing, it's going to continue to plug and plug and plug.
Why isn't D negative feedback?
D, if you scratch a rash and it spreads, a rash is not healthy, right? So normally we try to not keep rashes. So if the rash spreads, that's a positive thing, and when it itches even more than it did before.
I think I'm having trouble understanding like why negative feedback is different from positive feedback, like which one is which.
Negative feedback is when the variable is within, when you have it within a normal range. And when it deviates out a little bit, the signal immediately pushes it back into range. So we'll pretend that I have a range right here and right here. I have a variable that sits right here within that range. When it deviates, there's a stimulus that goes out. The integration center is involved. And the integration center sends an effector back and pushes it back into the normal range.
So would you consider, like, for instance, like earlier when you're talking about the blood vessels contracting and expanding, would that be considered a negative feedback system?
It is.
Because it's keeping that regulated to that certain temperature?
Correct.
And positive is just when something happens that it doesn't go back.
Yeah, until it keeps going out. The change gets bigger and bigger until whatever the stimulus is that's measuring the change, whatever is causing the change,
stops
away. Yes.
Okay.
Exactly.
So positive, it has to stop, and then negative, it keeps it in the range and it still stops. It just wants it to be at the same point that it started.
Yes. So what positive feedback, whatever change occurred, keeps going out more and keeps getting amplified bigger. The change keeps becoming bigger and bigger and bigger until whatever started it stops.
Okay, so that means for positive feedback is a reaction that is kind of obvious, right? Like it's seen. You can see whatever reaction is happening. Is that what it means?
Well, yes. Yes. Let me say yes, because positive is just going to keep getting bigger and bigger, and the more bigger it gets, the more obvious it's going to become. But initially, it's not very obvious. Does that make sense?
Yeah.
Can we have some more examples of positive?
Let me think for a second. You get an infection. Get a mild infection. And when you get that mild infection, your body puts out a response. What if that infection doesn't go away at that moment? The response gets bigger. The immune response that your immune system works to get stronger and stronger until the infection goes away.
So it can change between positive and negative? Because it kind of sounds like it went from positive and then switched to negative.
It will a positive feedback will keep getting bigger and bigger until whatever initially caused that change stops.
Okay.
So if like you had that infection and you had medication that helped stop it, would that make it a negative because you had an aid in it?
That would just help you to minimize that just helps your immune system.
Like speed up the process?
Yes.
Okay.
Because you having constant inflammation, if you've ever seen people who deal with chronic inflammation issues, that's not good for your body. So the antibiotic just speeds up that process for your body, rather than let you become immunocompromised, because your body keeps trying to fight it, but eventually your immune system is going to start struggling. Does that make sense? I mean, having my niece who just had 30 hours of labor to have a baby, they eventually stop. You got to do something about that. And she eventually had an emergency cesarean because your muscles are going to wear out at that point.
So just to use the example of the blood, and you said it was that negative feedback, or the blood temperature.
Okay, blood temperature. Okay, I was
Or
thinking.
not blood temperature, like the actual body temperature or whatever. Sorry.
Yes, you're fine.
So when it goes back and forth, why isn't it that these like, I'm sure there's something that is causing that to go back and forth. Why isn't that creating that to be a positive feedback system, since it's, because I mean, if your body just kept getting hotter and hotter when,
It will.
or colder and colder, I should say.
Because if your body keeps getting colder and colder, you're going to die. Shoot, let me think. If there, the best way I can say this is there's just certain systems, certain physiological symptoms that will, if they keep going out in a self-amplifying cycle, are going to be completely destructive. If
I guess maybe if like a negative feedback would be something that, if it were not to be fixed, would be have like a negative consequence.
Yes. Yes. If we continue, if we come back here, we continue to dilate, we get cold, it starts to constrict, we may block blood flow altogether. If we block blood flow altogether by constricting that vessel to the point of where blood can't flow through, we're going to have a real problem. Will you give me a day to think about this?
Oh yeah.
Give me a day to think about this, and I will come up with a better answer for you. Remind me about it too. I have a tendency lately where I tell you that I will revisit something, and then for some reason I can't remember. I'll think, okay, there was something I was supposed to think about, and I can't remember. Remind me about it. Midway through a 5-mile workout, a runner begins to sweat profusely. The sweat glands producing the sweat would be considered which part of the feedback loop?
Effector.
Yes. Homeostasis is constantly being disrupted. Physical insults, changes in the internal environment, physiological stress, mild or temporary disruption, and balance is quickly restored. Intense, prolonged destruction, poisoning, or severe infections. So, severe infections meaning not just a mild bacterial infection, but a massive systemic infection. The infection goes systemic, it's a positive feedback loop until the infection goes away. But when it goes systemic, you start blocking off other organ system functions. You start lowering blood pressure because you start changing things that can cause destruction of other organ systems. So a homeostatic imbalance are normal equilibrium of body processes that are regularly disrupted. Moderate imbalance is a disorder, meaning an abnormality of structure or function. A disease is a specific illness with recognizable signs and symptoms. A local disease affects one part or a limited region of the body. A systemic disease affects either the entire body or several parts. A local disease would be something like a sprained ankle, a broken pinky, a bruised rib, or a broken rib. Systemic disease is when that bruise or that broken rib impacts more than just that area. Symptoms are subjective changes such as a headache or nausea that cannot be observed by someone else. Signs are objective changes such as fever or swelling in which a clinician can observe and measure. So fever you can measure on somebody else. Fatigue you can't. Someone can say, I feel really tired. There's really not a way of measuring that one. A severe imbalance is death. So these three things, is fatigue a sign or a symptom?
A symptom?
A symptom. What about a headache?
Symptom.
Symptom. What about joint pain?
Symptom?
Yep. What about a blood test that's positive for Borrelia, a bacterial infection?
A sign?
It's a what?
Sign?
It's a sign. You can see the coagulation of the blood. A bullseye rash.
Sign.
The sign. And a tick bite, you pull out the tick, you can pretty much assume that it's biting you.
Sign?
And Bell's palsy.
Sign.
Suri goes to her family doctor and tells her that she has a headache and that she feels nauseated. The doctor checks Suri's temperature and finds that she has a fever. In the above situation, Suri's headache is a symptom of an illness. Why? While the fever is a sign of illness. Did I read that right? Go ahead.
True.
True.
Is the headache a symptom or a sign?
Symptom?
It's a symptom. So yes, that's true. Let's see here, gradient, difference in chemical concentration, charge, temperature, or pressure between 2 points. So a gradient is a change from a high to a change in the differences of a chemical concentration, difference in charge, difference in temperature, or pressure. One thing that I want you to remember all the time for the rest of your A&P studies is unless you put energy into something, everything flows from a high to a low. unless you physically go in and try to move it or push it one way or another, everything moves from a high to a low. Every interaction that we will see is going to move from a high to a low, unless we physically are doing something to prevent that or do something to create a gradient. Everything is going to move from a high to a low. I'm going to ask you this question because when we start talking about nerves and so forth, we're going to talk about ion gradients and electrical gradients. And if you can keep in the back of your mind every time that it's always moving from a high to a low, that will make your life a lot easier. Everything moves from a high to a low, every time. For example, blood flows from a higher blood pressure to a place of a lower blood pressure. If you want to create a gradient, you have to move things in the opposite direction or up the gradient. If you up the gradient, you increase the amount of change between those two areas. Movement in the direction to create a gradient requires spending metabolic energy. You have to put energy into it to make a gradient. If you don't have to put any energy into it, it's always going to move down the gradient. It's always going to go from a high to a low. So chemicals flow down concentration gradients. Charged particles flow down electrical gradients. And heat flows down thermal gradients. I don't want you to leave. I don't want you to, let me say this. I need to be able to take a role. To maintain your anonymity here, because some people do not want to be recorded. I don't want you to, let's see, how do I say that? I don't want you to leave. But let's take 5 minutes. You can mute your mic. You can block your video for me. You don't have to. You can if you want. But I need to make a list of names of who's here. So take 5 minutes. Just don't don't leave the Zoom, because that will make you absent from my attendance here. But walk away for a couple minutes and get up and stretch, and let's come back and start chapter 2. Is that clear what I'm asking you to do?
Can I please excuse myself quick to set up my son for school, please?
Of course you can.
Thank you.
Just don't leave. Keep your name on there.
I have a question for you. Do you teach A&P too?
You know what? I don't know right now. Being an adjunct.
Yeah.
I teach whatever they want me to teach. And so I might be.
Oh, okay.
If you ask me a little bit further in the semester, I'll be able to tell you.
Okay. Can you say if you do teach AMP2, are you gonna use the same book or?
We will be using the same book. I can almost guarantee that.
Okay.
Can I ask a question as well?
Of course.
So on the worksheet for chapter 1, it talks about like defining organ systems, organs, and tissues. Would that just be like organisms are composed of tissues, like that kind of definition?
Yeah. Read the question to me. Do you remember what it says?
It just says define the following terms: organ systems, organs, tissues, cells,
Yeah.
etc.
So yeah, like organ systems are
Composed of organs.
yeah.
Okay. I just wanted to make sure that's what you wanted.
Yeah, an organ system that performs specific functions.
Okay.
Or set of functions that work together. What I would probably ask you is I would ask you what's an example of an organ system, and I would give you like stomach. I would give you nervous system. I would give you endocrine system. No, wait. Let me put this differently. If I ask you what here is an organ, and I put down stomach, respiratory, cardiovascular, nervous, which one of those is an organ?
The stomach.
Right. If and then organ would be defined as one or more tissues coming together to form a specific function. Does that make sense?
Yeah.
For the components of the feedback, is stimuli one of them?
Say it one more time, Esther.
Oh, no, this is not Esther.
I'm sorry because I can't see his face.
That's okay.
Who is
Yeah, that is.
hi, Dallas?
For the components of the feedback loop, is stimuli one of them?
Say it one more time?
Stimuli?
Can you say it one more time?
Stimuli. Is that one of the components of the feedback?
It, according to your book, is not.
It's not? Okay. So it's just receptor, control center, and effector?
Yes.
Okay.
I personally, I think it would be a component. It depends on what book you look at.
Okay.
But I think it personally would be because that's what causes the change.
Yeah.
Danielle, as I will tell you, the reason I can't tell the difference between you and Esther is you sound very similar in a way, and I'm hearing impaired. And so when I'm trying to match voices with names, and sometimes I get so that I'm reasonably good at it. Sometimes I'm not. So I apologize for that. Guys, when you're ready, come back. Come back. So I'm going to start here. So the chemistry of life, chapter 2. This is something that's actually covered in chapter 1, and I did not get it in there. But I want to just talk really briefly here about the scientific method. When most of you are going into some form of care field, I don't know all the programs that HAC has set up, but a lot of you are going into nursing, maybe dental, maybe veterinarian, something to that effect, right? Is there anybody who's not going into that kind of field? Whether you realize it or not, every time that you see someone who is telling you that they're struggling with something, you see a patient who says, I don't feel well, you are going to be using the scientific method every time. It's not as complicated as sometimes we make it out to be. What if, for example, I'm going to try to call on someone that I don't see their faces here. Or Daniela, question for you. What are you going into?
Nursing.
Nursing. What kind of nurse do you want to be?
A registered nurse.
What do you want to practice? Do you want to practice family? Do you want to work with children? What do you want to do?
Labor and delivery.
Labor and delivery. Okay. So you have a lady who comes in, maybe like my cousin, who says that you haven't seen her before, and she comes in and she says, I've been having contractions. So she comes in, she's been having contractions. She may look really big, but does that really mean that she's that far along in her pregnancy?
Wait, say that again?
Even though she may look like she's really big,
Okay.
does that mean that she's really far along in her pregnancy?
No, she could still not be big and still be far along.
Right. So that's one observation that you've made with regards to the scientific method, where you know that she may look big, but that doesn't mean that she is that far along. That is what we call an induction, where you're using information that you know from the past, like the fact that she may look big, but she's really not that far along. It's a possibility. You're using what you know from the past to make determinations now. So she comes in, you generate a hypothesis, you start talking to her, you find out that I'm trying to remember what is it, I think it's 42 weeks. She's 42 weeks along and she's having contractions. What can you say at this point?
Well, she's having contractions, then you could go into labor, or?
Yeah, she's probably having the baby. At this point, and she's probably ready to be having contractions because she should be having that baby. Now, she should be having that baby. Now, she says that she's been having contractions for probably the last 30 hours, like my niece.
Okay.
What do you say at that point?
A what?
If she's been having contractions for 30 hours, what do you say at that point?
Maybe her water broke already, and she's very close.
Or there's something wrong. Maybe you look to see how far she's dilated. You look at different things to figure out just how far along she really is, right?
Yeah.
Because at that point you may have a problem. And you may need to hypothesize another proposal in your head. You start thinking, okay, well, maybe we need to rethink this and let's look at it another way. For all of you that are going into the healthcare field, you're going to be using the scientific method all the time, whether you realize it or not. The first thing you want to do, though, is make lots of the observations. Major part of science is you make lots of the observations. You're always making measurements. You want to do all the assessments you can to make the right hypotheses and make the right correct decisions for that moment. And then if your hypothesis is wrong, you can reassess. But you're going to use all of the observations you possibly can make before you come up with a proposal. So, as a short synopsis of that, you're going to spend a lot of time as a healthcare provider making hypotheses and testing them. And maybe you'll have to rule out that hypothesis and realize that it's been rejected by other observations and other tests that you performed and move on to another hypothesis until you figure out what's wrong with somebody, right? Or what's really going on. I think the longer you are in your studies and when you're out doing what you want to do because you've suffered all the way through school, I think the more and more you look back on the scientific method, if you study it while you are doing your analyses and looking at patients, you will realize how much you actually use the scientific method. I have my roommate that I live with is a retired nurse practitioner, and I have several siblings that are nurses and PAs and so forth. You're going to use it more than you ever realize. Understanding biochemistry is essential to understanding anatomy and physiology. Biochemistry is the study of molecules or matter that make up living organisms. Anything that occupies space and has mass is matter. It can exist in 3 different states: solid, liquid, and gas. You guys, if this chapter is complicated for you and if it's a difficult chapter for you, I need you to go back and look at your first biology course that you took. How many of you here at Hack, what was your first biology course before
This
this?
is my first.
I took one last semester.
What did you take last semester?
It was something like this, but it's supposed to be like a biology class, like from high school, but it wasn't like high school. It was something more like this.
I need you to really study this chapter. If what we go over in this chapter is not a review of what you've seen before, I need you to really study this chapter. If you study it really well, you will save yourself a lot of grief and me a lot of grief over the next semester to year. You'll save yourself a lot of grief over the year, and you'll save me a lot of grief and yourself a lot of grief over the semester. Because this is the basis of all physiology. So most matter in the body there's 4 types of biomolecules composed of chemical elements. There's carbohydrates, fats or lipids, proteins or nucleic acids. Sorry, my cat just jumped up into my lap here. Chemical, you're gonna show your face. I told you guys about my cat that ended up getting down to 6.5 pounds.
Yeah.
This is that cat. He says, I'm now at a very healthy 11. Huh. Yeah.
He's cute.
He's cute. He's my little monster. He's a Maine Coon mix. And if you know anything about Maine Coons, if you look at him, you see the little tufts coming out of his ears. And he says, Mom, I really hate being on show and tell. But he's very fiery. He gets very mad at me really quickly. Josh was talking about eye dilation and the sympathetic nervous system the first day. This cat, I can make him mad really, really fast. And I can upset him really, really quickly, and then he burns down just as quickly. The second I stop harassing him, he's fine. But I like to make him mad sometimes just because it's fun, because it happens so fast. So chemical elements are the unique substances that cannot be broken down into simpler substances by normal chemical means. The 6 major elements. Leo, don't do that. 6 major elements that make up 99% of the human body are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. Good way to remember this is the mnemonic schnapps. C is in Charlie, 2 C's, C is in Charlie, C is in Charlie, H is in hotel, N is in November, O is in Oscar, P is in Papa. So one of your questions asks you about the 6 elements that make up most of 99% of the body. That's them. So the majority of it is oxygen, then carbon, hydrogen, nitrogen, calcium,
Phosphorus.
phosphorus, correct. Some lesser ones, sulfur is a pretty high one, chlorine is a pretty high one. Potassium and sodium, these are very vital, minor, smaller ions. That's why they don't make up such a big mass. But are they vital to homeostasis? Yes, they are. Especially potassium, sodium, and chlorine. Several of them elements, why is this, I want to move this a little bit. I wish I could move my screen. Several of the elements that make up the human body are minerals or inorganic elements that must be taken in via your diet. 75, and they make up approximately 4% of the human body weight. Calcium and phosphate are 75% of those minerals. The other 25% are chloride, magnesium, potassium, sodium, and sulfur. Major functions of minerals is that we have crystallized calcium, phosphate, magnesium, fluoride, and sulfur ions in the bones and teeth, make up the rigid structures of the bones and teeth. Proteins commonly contain sulfur. We'll talk about the amino acid cysteine probably on Monday. Not Monday, I'm sorry, next Tuesday. Contains sulfur. Phosphate, major component of nucleic acids, ATP, and cell membranes. We'll talk about phospholipids. Called phospholipids because one of their major components is phosphate. Enzymes and other organic molecules require minerals to function. They require them within their structure to maintain their structure. If you remember, we talked about complementarity in chapter 1. To maintain an enzyme structure, it sometimes requires a mineral such as magnesium to maintain the structure of that enzyme. And if you can't maintain the structure of the enzyme, this enzyme will not function right because the function is reliant on the structure. Atoms are building blocks of elements. Atoms are composed of subatomic particles. There are 2 different models that are used to describe atoms. The first one is the electron cloud model. The second one is the Bohr model, or the planetary model. Planetary meaning it's arranged like planets around the sun. More typically, an atom looks more like this. If you look at this little picture that I have here, do you see that it's darker? There's a ring, it's lighter right around the nucleus of the atom. It's a little darker right here. The reason for that is with the electron cloud, electrons can be anywhere at a given place, but they are more likely to be in certain areas more than others. The possibilities of them being in certain areas are higher than them being in other areas. When I refer to these areas, I'm referring to orbits. They're more likely to be within a given orbit than they are be in between the orbits. But at any given moment, we cannot detect exactly where an electron is. But we can say that they are more likely to be in the orbits than they are to be in between the orbits. So it's a matter of averages. Where is it more likely to be? On the average, where is it more likely to be? So electrons forming the electron cloud in an atom occupy regions of space called electron shells. And each shell contains one or more orbitals.
On the outside?
Yes.
Surrounding it?
Yes. So we refer to different shells, electron shells, but within each shell, going from the inside out, each one of these shells can have more than one orbitals. For example, we can have one that's going this way. In this first one, there's actually only one orbital. So let me go with another one here. In this outer orbital, we can have one that's going this way around the nucleus. And we can have another one that's going this way around the nucleus. They're both within the same shell. They're both the same distance away from the nucleus. Neutrons are neutral in their charge. Protons are positive in their charge. Electrons are negative in their charge. One thing that I want you to keep in the back of your head, because we're almost done here for today, is do like charges, positive and a negative charge, do they attract or do they repel?
Positive, like charges repel.
Say it one more time.
Like charges repel, unlike charges attract.
Okay, so there has to be a certain amount of energy in each one of these shells that prevents the electron from flying into the proton. There has to be a certain amount of energy, sorry guys, there has to be a certain amount of energy that maintains this distance. The greater the distance, so from here to here, the higher the energy required to keep the electron from falling into the protons. Keep that in the back of your head between now and next Tuesday. But I'm going to move forward a little bit more. But I want you to keep that in the back of your head because we're going to talk about it in a little while.
You said the energy increases to keep them apart?
Yeah,
correct?
keep,
Okay.
the farther apart they are, the more energy that's required here to keep them apart. Actually, wait, let me say this differently. The closer they are, here, when it's relatively close, the further they are here, it's going to go from being high here, medium here, but the further you get, the less pull there's going to be. It's kind of like working with a magnet. Initially, that pull is really strong. It's fairly strong out here. The further you get, the less pull there's going to be.
So the closer, the higher the energy, and the further, the lower the energy? Is that what you're saying?
In the middle, it's the most intense. And the further away, the less intense it's going to get. I will try to put that a different way on next Tuesday. But right in the middle, we're going to see that these electrons can absorb energy. And right here, this electron can absorb energy and move up to this orbital. It can fall back down and release that energy. But the further away, on top of that, in addition to that, the further away that electron gets from the nucleus, the more likely it is to react with other atoms. So the further away it gets, the more likely it is to interact with other electrons in other atoms. Right now, right here, the pull is strong. Right here, this electron can gain a certain amount of energy and move out. And then it can move out further. It has to overcome the pull here to move out. It has to gain some energy to overcome the pull here. I'm not explaining this very well, and I can tell. Give me a couple days to think about this one.
Are you trying to say like kind of like you're holding, let's say you're holding the magnets together and they're being pulled together because one's positive and one's negative?
Correct.
The closer that you get the magnets, the harder it is to keep them separated. So you're using more energy to keep them separated than it is the farther you hold them apart.
Yes.
And
to move it even farther away, to move it even farther away, Josh,
yes.
I have to add more energy into it. But once it's at a certain distance away, the further, if there's another orbital out here, the further it is away, the less that pull becomes.
Yeah.
Yeah, that's a good way to explain that. I need to remember that one. So consider it like magnets that are orbiting. There's a magnet orbiting or a piece of metal that's orbiting this. I like scientific little things, so I've got my little model here. This one up here is more likely, maybe, to get pulled away. The pull isn't as strong because it's got to go through all the other ones to get there. This one down here, very bottom, that pull is a lot stronger for me to move it away. If I had something else like this one would be more likely to react with another one than this one down here.
Wait, the closer they are, the more likely they are to Or interact with each other?
The more energy is required to keep them separated.
The closer they are.
Yes.
They need more energy to keep them separated.
And to move it out further requires more energy. Kind of like Josh, explain what you were saying one more time.
Oh, like, so if you're holding two magnets together, think of like how much your energy to keep them apart is kind of like the energy to keep the atoms apart, so that you're producing in your hands. So when you're pulling your hands apart, it's a little bit more, it's not going to be as hard or as difficult to keep them apart. You're expending less energy to keep them apart. The closer you get them, the more energy you have to use to keep them separated, because the pull is going to be greater.
But then the farther away, the less energy you need to keep them farther away?
Yeah, so like if you literally take two magnets in your hand, if you have like two kitchen magnets on your fridge or something, and you put them together, and when you first pull them apart, you have to use a lot of energy, and then it gets real easy because the force between them isn't strong enough to pull, and you don't feel it. So you get out to the end, like when your arms separate, you don't feel it. You're not using any energy to keep them there. When you're getting closer and getting touchier, you have to use some energy to keep them from just bumping together. Does it make sense?
Yeah.
Like you can feel the energy in your, like as your muscles are creating energy to keep them apart.
Keep them together.
Kept thinking that our class finishes at 11:15, but it actually finishes at 11:45.
Oh, I thought you were gonna let us out early.
Let's not do that, because it just means that there's more next time.
Yeah.
I kept thinking, no, it's not 11:15. Smallest particles of an element that still exhibit the properties of an element is the definition of an atom. The smallest particles of an element that still exhibit the chemical properties of that element. So the smallest part of an element that still exhibits all the properties of that element. Elements can be arranged on the periodic table. Bear with me, I don't know why, I don't really like how this screen works. I'm gonna try something really quick. Would like to zoom in a little bit so we can actually see the whole slide.
I think if you pull your bar down, that's on the far right of your screen, so we have just the top of the picture, you can see more of it, because you can only see half of your image.
Pull down.
To the upper right of the picture, if
Yeah,
you hit
right
the
there.
4 arrows. No, to the right of the picture. You'll see 4 arrows that are all going away from each other. Right beside your mouse to the right. Right there.
Right where all the little strip of faces were. Okay, let's do
If
that.
you maximize that, it'll make it bigger.
Okay, let's do. I apologize, guys, because I was like, this is
That's okay.
not great. So, periodic table. Elements are arranged on the periodic table according to their atomic structures, which results in their different chemical properties. The grouping of the different elements shows if they're grouped closer together, the more similar their atom structure is. The more similar their chemical properties are. Elements over here, their atomic structure is more similar. Their chemical properties are more similar. Hydrogen is very similar to lithium in its chemical properties. Fluoride and chlorine, very similar in their chemical properties. The atomic number of an atom or an element is the number of protons within an atom. For example, for hydrogen, it's 1 because there's only 1 proton. In stable atoms, the number of protons equals the number of electrons. Stable atoms meaning they're not very reactive. They're not as reactive as others. The number of electrons determines the chemical behavior of the atom. How easily does it react with other atoms? The mass number is the sum of the number of protons and the neutrons. For the case of hydrogen, the mass number is 1 because there are no neutrons. Nevertheless, for isotopes, which are atoms with the same number of protons and electrons, there are different numbers of neutrons. For example, deuterium down here contains 1 neutron. Stable isotope for hydrogen contains 1 proton, 1 electron. Deuterium, which is an isotope of hydrogen, contains 1 neutron. Tritium contains 2 neutrons. So isotopes of atoms are atoms with isotopes of elements, excuse me, let me say this right. Isotopes of elements are atoms with the same number of protons and electrons, but a different number of neutrons. The atomic weight is the average relative weight of all the isotopes of a given element. So we're talking about protons, neutrons, and electrons. Even though we can't physically measure them, they all have a certain amount of mass. They all have a certain amount of weight. The atomic weight is the average of all the weights of the mass number, meaning the number of protons and neutrons. For hydrogen, the average for all the isotopes is approximately still 1. Because there's only 1 proton. Questions about this?
So as it goes up in the scale, like, does that get closer to like what the weight is? Like helium, would helium be 2?
Yes.
And then so on and so forth?
Yes.
And I'm assuming this is just rounded. I'm assuming these don't have like the decibels and stuff.
No, they don't. But you'll see that they deviate a little bit as you go farther out. Initially, that's the way it is, but they deviate a little further as you go out.
I'm sure with all the rounding off that they do, they catch up at the end.
As lithium 3 and 7.
Yep. So what is the 3 up top is telling us the atomic weight? What is the 7 telling us?
The 3 is telling you how many protons are in that. The 7 below is telling you on average how many neutrons and protons are there. There's on average there's maybe there's a little bit more neutrons there. So if I take 3 divided by 7, it's 3.5, right? Or no, sorry, 3, bad.
Wait,
It would be 2.3 repeating.
Yes. Let me say this differently.
the 7 is protons and neutrons?
3 divided by 7, or 7 into 3 is what?
2.3 repeating.
I'm not saying this right.
Wait, 7 into 3? Are you're saying like 3 divided by 7?
7 into 3. What is that?
2.3.
If you're saying it's 7 into 3, it's
How many times does 3 go into 7?
2 point like 2.
2.3.
2 times. 2 times. Why am my brain is I'm having a brain moment. Give me one moment here. 7 is typically in lithium, there is in a regular atom that is not an isotope, there is 3 protons and 3 neutrons. However, there is an isotope of lithium that contains more than 3 neutrons. That's why the atomic weight is 7 and not 6. So the 3 there.
It's telling you the number of protons and neutrons if it's not an isotope.
Right. So
and then the 7 is telling us the weight of all that, which would be.
It's telling you that there's an isotope out there that contains one more neutron every once in a while. It's not as common as the regular atom.
So is the top protons or protons plus neutrons? Because you guys said both, so I'm confused.
Sorry, the top one is protons.
The top is protons, and then the bottom is the atomic weight?
Yes.
Okay.
Because the top's protons and we're not doing an isotope, we're just adding the 3 plus the 3. But you're saying that there's one there with lithium, there's a possibility. So let's say you go right to the next one. I don't know what BE stands for. There's 4 protons in that, and there's 4 neutrons, which will give you 8. But there's also a variable that has 1 additional neutron.
Correct. Josh, you're good at explaining this stuff. You should be teaching. I'm sorry.
I'm terrible.
that made sense to me. So, for example, we're looking at. I gotta remember what this is. Psyllium. See, it's not psyllium. Ciliate.
Silicon. I guess.
Silicon. Thank you. 1428. There's not a lot of isotopes there's not any isotopes almost of silicon.
Same thing with magnesium, or I'm assuming magnesium is 12 and 24. It's always going to be, there is no isotope for that.
There is, but it's a very small amount. Remember, there's 2 for hydrogen, but it's still 1 and 1.
Correct.
Or that actually says 2 there. But we know that the average weight is 1. This is not an exact periodic table. The key of this periodic table that I wanted you to get was that elements are on the periodic table according to their chemistry.
All right.
Let's see here. So radioisotopes. Remember, now we're just talking about isotopes that are now radioactive. Radioisotopes are heavier isotopes of elements that are unstable. So the heavier the isotope, meaning the heavier the number of neutrons and protons there, the more unstable it is. Their atoms decompose spontaneously to form into more stable atoms. The process of atomic decay is called radioactivity. So the process of this heavier isotope of an atom spontaneously decomposing to form a more stable atom, that decomposition is radioactivity. The decay involves the emission of alpha, beta, and gamma rays from the atomic nucleus. So when an alpha ray is emitted, the daughter nucleus loses 1 helium nucleus, which is 2 protons and 2 neutrons, and an alpha ray of energy. Actually, I don't think that's correct. Let me say this differently. The alpha ray there is the helium nucleus. Maybe.
So that's when you're gaining protons and neutrons?
You're releasing them. Radioactivity is the release of it. So this is when this nucleus right here, the result is that you get a more stable nucleus. You get the release of this. For this to become a more stable nucleus, it has to give up this.
Are these just 3 different forms of radiation? Is what?
Yes.
All right.
And it depends on what isotope it is and what type of radiation it's going to give up.
And that depends on what it's used for.
Or what's the atomic structure. It's specific to the element.
So basically, you're also saying that all elements want to go back to being, what was the word? More stable. I said more stable. So they're always looking for some way to go back to their stability. So they're just going to find one of these ways.
Yes. Being unstable, physically, they can't be that way. They're going to work to become more stable. Just like, think of it, you know how I was telling you that in the back of your head, I want you to keep in mind all the time that things go from a high to a low.
Yeah.
Something that is high energy is going to move towards a lower energy all the time. Go ahead.
I have a different question, but you can finish your.
No, please, go ahead.
So like we're supposed to define alpha, gamma, and beta rays. What would we describe each of those as?
It is this. A helium nucleus is an alpha, an electron is a beta, and a photon is a gamma. More than anything with regards to those defining those rays, I'm going to ask you, one of my next questions has to do with their intensity. What are they used for? What's the most dangerous type? What's the weakest type?
So you would just
Go ahead.
a silly question. Instead of the plus signs, they should actually be minuses because that's all those are what you're taking away, or just the first one is what you take away and the other ones you add.
Let's look at this as being a chemical reaction. This breaks down to be this and this.
Okay. So those will equal that original.
Yes.
Okay.
Yes.
Does it divide into all three every time, or it becomes one of those three options?
It's going to become one of those three.
Okay.
It depends on what kind of chemical element it is, which one it will become. But yes, it's going to become one of those three. Like we are going to learn, and you probably already know, you may already know this, hydrogen isotopes are not that dangerous. Radium isotopes are extremely dangerous based on what they emit when they decay. I've worked with tritium before. It's not that dangerous. I don't think I'd ever want to work with radium because it's a lot more dangerous based on what it releases when it decays. Have you ever heard? And I like watching NCIS, and I like shows like that. If you ever heard of someone who's dying of radiation poison, they refer to them as the Walking Dead because there's really no way to save them at that point, and it's a cascade of damage. And I got that from the show NCIS. One of the titles of the shows is The Walking Dead. So radioisotopes gradually lose their radioactive behavior as they become more stable atoms. The time required for a radioisotope to lose 1/2 of its radioactivity and become a more stable atom is its physical half-life. So we have a beaker of radium that we've got in a very nice protected thing so we don't all die of radiation poisoning. The time that's required for half of it to become a more stable atom is its physical half-life. Half-lives can vary between hours to thousands of years. With regards to medicine and research, the biological half-life is how long a radioisotope will be how long will half of the how long will the radioisotope be radioactive within the body? I don't think that's right. Bear with me just for a second. It just doesn't sound right. Okay, so similar to the physical half-life, the biological half-life is how long will half of that radioisotope be? How long will it be until half of that radioisotope is no longer radioactive within the body? How long before half of it decomposes?
1 year.
It depends on the isotope.
Oh, I thought you were looking at the one in the graph. Sorry.
No, it's okay. This is a biological one.
Okay.
Sorry, this is a physical one. Wow. This is a physical one. This is for cobalt 60. And it tells you that the half-life for is about 5.27 years. So if you go ahead.
No, you're good.
If you put this into someone's body, if someone ingests a bunch of cobalt 60, I wouldn't recommend it. Within 5.27 years, half of that should be gone. And it should no longer be, it should be a stable atom at that point and no longer be radioactive. Radioisotopes are tools for biological research and medicine. If you remember with the PET scan, do you remember how the PET scan worked? The positron emission tomography. If you remember that, what we did is you inject a radioactive glucose tracer and you're measuring how much radiation is coming off it. And that's how you measure the breakdown of the glucose. How much of it is being utilized of that? So radioactive isotopes share the same chemistry as their more stable isotopes. They can be used to localize and eliminate damaged or cancerous tissues. This is an example of Graves' disease, which causes hyperthyroidism. So too much activity in the thyroid gland. From what I understand, the way that they would do this is they would give someone on radioactive iodine to see how much of the thyroid is, how quickly is the thyroid using up that iodine. And if it lights up like this with radiation, in comparison to this in a normal individual, that's a sign of hyperthyroidism. It's being used up way too fast. So radioisotopes are tools for biological research and medicine. So when someone asked me about defining alpha rays, gamma rays, and ionizing radiation, and your name wasn't here, your face wasn't here, so I don't know. I apologize if I don't know your name. The alpha emission is the least penetrating, therefore it's the least damaging. So if you remember from up here, that's this. It's the least damaging. gamma emission, like radium and like cobalt 60, has the greatest penetrating power. It's used to destroy local cancers. So this would be what someone would be treated with when they're undergoing radiation for cancer. That's a really good question right there. This is what you'd be using to treat cancers with.
Would they use any of the alpha, like, is alpha?
No, because it's not as that would be it's not going to work as quickly. It's going to cause irritation, because if you see here, one example of smoking is the inhalation of alpha particles from radon or uranium decay, which can cause lung cancer from just the regular irritation. One thing that I hope you learn, and I don't know if we'll talk about it this semester or next semester, but chronic inflammation, like from smoking and constantly inhaling things that are foreign into your body, can result in cancer. Your body is constantly trying to repair itself, so eventually something is going to go awry. Your body is constantly trying to fight something, so eventually something is going to happen where a cancer is going to develop. So alpha emission it's not immediate enough to be very useful in cancer. Ionizing radiation or beta rays is where electrons are knocked out of atoms, and these electrons go on to destroy other things. If you remember, we were back here. I'm sliding back. Bear with me. If you add electrons onto atoms that don't normally have that many atoms, you're going to change their chemistry. And if you change their chemistry, they may not do what you need them to do anymore. And when they don't do what you need them to do anymore, you've got a loss of function that can cause problems. So ionizing radiation knocks electrons out of atoms, and these electrons can go on to destroy or injure other things. So it's a more, what's the word for it? It's a more, it's not as penetrating, but it can also cause a lot of damage over time. Chronic exposure to this is probably worse than localized exposure here. I'm not an expert on radiation. I'm telling you how I think it probably works there. Because if gamma emission caused this kind of radiation, we would never use it on cancers. But they localize this, they put down a shield to block it in other areas to specifically only treat that region. Okay, you guys, now it is 11:51. Let's end class here. I'm sorry I went over. Feel free to tell me when it's 11:45. Are there any questions?
Um, I don't have a question per se,
Go
but
ahead.
I think regarding the periodic table, for a better understanding, we have to like know the characteristics of
No, no,
um
no,
atom.
no. Bear with, Esther, over the next day, you will become more familiar with how that works, how the periodic table works. I
Yeah,
will,
because
I want, more than anything, I want you to understand patterns.
now I was just looking at the periodic table for beryllium. So it seems like the atomic number, the
Yeah.
one up is 4, and the atomic mass is 9. So that means the number of electrons in the shell is going to be 2 and 2.
No,
I mean up here for beryllium, the atomic mass is 4.
this one right here,
Yeah, I think the 4 is the
there,
atomic mass,
there's
the atomic number. So why the 9 is the atomic mass?
there can be an extra neutron in there from time to time. So this is 4 times the atomic, excuse me, the
Atomic mass.
atomic mass is usually 4 protons plus 4 neutrons, which would be 8. But on average, there are some isotopes of beryllium which add an extra neutron in there from time to time. So the average atomic weight is 9. Because there are isotopes of beryllium. For ones like carbon, where there is 6 and 12, there's not a lot of the amount of isotopes are not very common in comparison to the stable atom.
So for carbon, the atomic number is 6.
Yes. So there's 6 protons there. And on average, there's 6 protons and 6 neutrons, and that's what that 12 means.
How do you know how many electrons it has?
You can determine, with very few exceptions, you can say that the number of protons equals the number of electrons.
The number, so if it has 6 protons, it has 6 electrons?
Correct. But we're going to talk about some exceptions to that next time. We're going to talk about ions, where we can give up electrons. But right now, yes.
For like a standardized all non-isotopes.
An atom that doesn't have any charge on it, the protons equal the number of electrons, because the number of pluses and the number of negatives equal.
Okay.
And these are just based off neutral atoms.
Yes.
Just so you have like a base.
Yes. If the number of positives and the number of negatives equal within that atom, which is what we see here on the periodic table, that is the charge is 0. For an ion, which we'll talk about next week, that's when you add or subtract electrons away from the atom.
Yeah.
And that's going to make the overall charge on the atom positive or negative. That's an ion.
Yeah, so these are just atoms at their rawest form.
Exactly. These are their neutral form.
Like when you talk about ions, that's adding stuff and creating change to them.
Yes. And you guys, because I went over and I don't know how many students actually have classes right after this, and I apologize for that. I will start right I'll start about right here.
Okay.
Wait, how do you know if it's an isotope?
What?
I'm confused about with the isotopes.
Hydrogen is unique in that it only has one. Wait, say it again. What's your question?
Isotopes, I'm confused about that whole thing.
I
It,
think
isotopes are when the number of neutrons is more than the number of protons within that atom. Normally it's equal. However, when it has more neutrons than it does protons, that's an isotope.
So like lithium will be an isotope? Because it has more
It has some
neutrons?
isotopes. It has some isotopes.
No?
There's stable lithium, and then there's isotopic. There's lithium isotopes.
I think a way that it makes sense to me is when I think of it is that these are all like the base. An isotope is just a variation slightly by adding a little bit of the neutron to create like a heavier mass.
Mhm.
And
So how do you know if it has more neutrons and it makes it an isotope?
If you look,
it will label it.
they have the same number of protons and electrons.
if you look here, magnesium there,
Yeah,
12 and 24. If
yeah.
you can divide 12 into 24 evenly, like right there, where it's half and half,
Yeah.
there are not a lot of isotopes of magnesium. There are almost none.
Yeah, it's just like it's saying that magnesium is the form and it doesn't have a higher number. The number is not larger because there's nothing that amounts to anything more.
Yeah, if there isn't enough that it's actually measurable for the most.
If you go to, for instance, zinc, there's 5, so there's a larger area of isotopes, so there could be more possibilities.
Okay.
Correct?
Yes. Yeah, I would correct you if you were wrong.
Oh, sorry.
You're fine. Yeah. I would disagree with you if you're wrong, but yes, you're right. Look at it that way.
It's think of the word as isotope as like a variation of
Yes.
the zinc. It just there's like 5 variations, and a variation of it is having an additional neutron.
Yes.
But it is still the same thing because the neutron doesn't actually change the charge of the what the charge is like the charge isn't changing. The neutron just is there, and the only thing that changes is the heaviness, like the weight of the.
Weight of the nucleus.
Yeah, that's true.
Right.
So it's the same exact thing. It's just like I don't say it's more. Well, it is going to be more dense because it has a larger weight.
Yes. Because remember we talked about down here with what you're saying there, Josh. Oh shit. Let me come up here. I missed it. Heavier isotopes. It's based
Yeah.
on the heaviness of the nucleus.
So if you were to get like an isotope that has like a lot more range of neutrons, the heavier that isotope is, the more like, let's say if you just took the regular, let me pull it up real quick. A regular, if you look at the bottom one, like the bottom line on the left, the FR.
Yes.
If you're looking at the 87, it would actually be, what is that, 160, 174.
Yes.
So if you're from the farther you get from 174 to 223, the more radioactive that isotope is going to be.
Yes. I
Because it's going to be
want to.
heavier.
Where's uranium on here?
Uranium, I believe, is
Right here.
Oh, right where?
what's the number?
Rhenium is, I think, 226. I'm not sure.
The bottom one. Okay. Because when you label across, you want to, if you reference an element, you always want to use the proton to reference the number, because that's your base.
Because it's easy to organize it accordingly.
Yeah, it's going to be a smaller number, and it keeps it more compact.
Okay, where's uranium on there, guys? You can. I don't have it memorized. Nobody has a periodic table.
Yeah, no.
Rhenium is 186.
186.
The Re.
All right, so 75.
That's
75. Okay.
yeah.
You want to
75?
yes. Is RE
Yeah.
uranium?
Re, yeah.
I thought it was you. But
Yeah.
let's go with that. Look at how 85 to 186 has a lot more isotope possibilities. Plutonium right here.
That will make it a radioactive isotope.
It could make it. It could.
Yes.
Okay.
If it's closer to just being even to like the 75 plus the one is 150, the closer you get to the 150, the less radioactive it's going to be. The higher you're going to get away from that, you're causing that weight, which is causing the instability, which is going to create more radioactivity.
You guys, I am not going to ask you what atom has the highest amount of possibilities for radiation. Not going to ask you that question. What I want you to know more than anything is how radiation works. What are the emissions? Because in a world that we live in right now, where there are certain people who are get higher levels of radiation than others, you need to be comfortable with the terms. But I'm not going to ask you what element is more likely to be radioactive than another one. I may give you some examples, like when we talk about bones here in 2 chapters, we're going to talk about radium poisoning, which was a major issue for a lot of people when radium was in paint. Radon was in paint.
So are you going to ask us the number of electrons per shell?
No, but I will explain that next time. I will possibly ask you that, but I'm going to explain it to you first.
Okay.
I will tell you it's 2 and then it's 8 for all the others.
Okay.
It's 2 for the first shell, 8 for all the others. And I will explain that to you because that's going to be important to how they react, how different atoms react. If they don't maintain 8 electrons in their outer shell, they're much more likely to react with other atoms than if they had 8. Before, Esther, to answer your question rather than hold it off for a whole entire week, look up the octet rule. O is in Oscar, C is in Charlie, T is in Tango, E is in Echo, T is in Tango rule.
Can you repeat that again?
Of course. O is in Oscar, C is in Charlie, T is in Tango, E is in Echo, T is in Tango. What that means is when you think of an octagon, what is an octagon?
8 parts.
It has 8 parts. So the octet rule is the rule that says that in the most outer shell, there has to be 8 electrons.
Okay.
If there's more or less than that, that atom is going to react more easily with other atoms. If it has 8 electrons or 2 electrons, look, let me show you here. Look here, this last row. Helium has 2 protons, 2 electrons, because the number of electrons equals the number of protons.
Yeah.
It only, in the first shell of the first electron shell, there are 2 electrons, and that's all it needs. Any shell beyond there, circling out from the nucleus, there's going to be 8 electrons in any of the other shells. There needs to be 8. So 2 in the first one, 8 in the next one. So 10. All of these are called I have to remember, the noble gases, because they don't react with other things. As a rule, they don't react with other things. They're not reactive at all. And the reason is because all of their outer shells are full. In that first shell, the helium, two electrons in the outer shell. It only has one shell, and it's full. Neon, there's 2 electrons in the inner shell. There's 8 in the next to equal 10. It's full. It's not going to react with anything. Argon, there's 18, so there's 2 in the first shell, there's 8 in the next shell, and there's 8 in the next shell. It's not going to react with anything. It's the same for all of those gases.
It's the only row where if you add 8 the whole way down, it's by 8 or by 8.
Yeah, 2 plus 8. 2 plus 8, because the first shell only contains 2. So if I look here, sorry, Bill, I should have scanned up. The first one only contains 2.
Mm-hmm.
Carbon contains 4 in its outer shell.
Yeah.
Which means that to be stable and not react with, this is the carbon atom. To be stable, it has to have 4 more there in the outer shell, or it will not be stable. So it is able to interact with up to 4 other atoms to become a stable thing. Everything wants to be more stable.
And it can't, and carbon's carbon because it can't have 4 more, because that means it would have more protons than neutrons, which would change it.
Yeah.
To be stable by itself.
We're going to find that we really, really like carbon this semester. And we like carbon because of its ability to make 4 bonds. It's very flexible. It is the basis for all physiological materials. I actually, for one of my classes that I'm teaching, now that I'm thinking about it, I wish I still had a organic chemistry kit here. But I think for one of the schools that I teach at, I'm going to go in and pick up a kit. I'll bet you it's in there.
Okay.
But just realize that if it has, the shells are filled, like 2 and 8 and 8 and 8, it's not going to react with anything. If it's not 8 electrons in these outer shells, it's going to be more reactive. And it's going to react with other things.
So wait, if there's like 2 in the first shell, and then let's say there's 6 Yeah, the shell. It's gonna react.
And if it's got only 6 in that outer shell, it's going to want to take on 2 more electrons
Yeah,
Okay.
because it wants to have 8.
that's true.
And it's going to do that through either sharing them or
From
taking
another
them,
atom.
yes, either
Okay.
sharing them with another atom, which makes a covalent bond,
Okay.
or it's going to take it to make an ionic bond
Where is it taken from?
from another atom.
Oh,
Work to wrap your head around this stuff. If it's not clear, work to wrap your head around it and be patient with yourself and realize that between now and next week, I really hope it will be a lot clearer. I mean, if you looked at this PowerPoint, it's 81 slides. It's a big chapter.
I know I printed it.
Be patient with yourself as we work through this.
So, just a reminder about what we discussed at the beginning of the class, the chapter 1.
Uh-huh.
You said you were going to rectify that.
I will have to actually talk with someone and figure out where I made the mistake and how to fix it.
All right,
So
thank you.
it's going to take me probably more than a day. Just be patient with me and let me by next Monday, if I don't have it fixed, feel free to harass me a lot. Or next Tuesday, feel free to harass me a lot. Okay? Get
All right,
on
thank
me
you.
if I don't get it done before then, okay?
All right, thank you.
Okay. You're welcome. Have a good rest of your day, okay?
You as well.
Okay, thank you. Oh, we could put white and right here. We're almost going to but I don't remember that being much. What I need to do is make a goal to start with there. And then I'll get going on my two. Josh is. Oh, yeah, you are. Oh. It's hard because you record them. Emma, are you there? Ali, are you there?